Hello to everyone, and welcome to today's webinar, Optical Fractionator to Quantify the Number of Cells. My name is Dan Peruzzi, and I'm a research liaison here at MBF Bioscience. I have been for about 12 years, and my job is to help you understand how you can use unbiased stereology to get the data that you need to answer the questions that you're asking in your experiments. I'm joined today by Julie Keefe. She is a product manager, and she has performed the optical fractionator many times, is an expert in it, and she's going to be helping me to field some of the more practical questions. This is a breakdown of today's schedule. For the first half, roughly 20 minutes on the theory, and then we'll break for questions. And then I am going to show a demonstration using a microscope of how to do the optical fractionator to estimate the number of neurons in a given anatomical area. And then we'll stop for questions. So the theory, the first part of the webinar, is going to have to do with how does the optical fractionator fit in with the other unbiased stereology probes? What are the rules we need to follow to avoid overcounting cells? And how much should we sample in order to get a good snapshot of what's going on and tease out any differences there might be between the means of the experimental and the control group? Then we'll pause for questions. Then we'll get to the practical demonstration for the second half of the webinar. I'm going to demonstrate how to estimate the number of cells using the optical fractionator workflow using MBF Biosciences software, Stereo Investigator. The reason I am using the optical fractionator is because it's the best and most reliable process for estimating the number of cells. This is a rigorous process. I am about to go over the set rules that we're going to use to do this. These are accepted rules. You can publish these rules in your papers. We also strongly suggest you publish 1 or 2 or 3 paragraphs about how you define your region of interest. You'll end up with a reproducible experiment that people can verify. There are other methods out there, such as setting a threshold at low magnification to detect the number of cell pieces. That's not what this webinar is about today. This webinar is about estimating the number of actual cells. And then we'll stop for questions after that. So let's start off with kind of an idea of what unbiased stereology involves. It involves generating virtual geometrical shapes which are superimposed by our program Stereo Investigator on images of the tissue that you're trying to find out about. So we're probing the tissue with virtual geometric shapes. Our job is to record the number of interactions between the virtual geometric shapes and the tissue being examined. Those are called events. The number of events is used in a formula to arrive at an estimate. So let's see what I mean by this. In this slide, I have drawn the tissue, the real tissue that we're trying to find out about, in green, and the virtual geometrical shapes are in red. So we can see here, we can estimate the volume of an anatomical region. An example might be the inferior colliculus or the hippocampus or any of their subdivisions. We can use points to estimate the volume. The more points there are on the cross sections through the volume, the more volume there is. We can use virtually created geometrical shape, a plane, which is shown here in red, to estimate the length of biological filaments, for example, blood vessels or nerve fibers. The event in this case is the intersection of the virtual plane with, for instance, a blood vessel. The more intersections there are, the more length there is. The smaller the probe that you are probing with, in this case the plane, given the same number of intersections, the more length that there is there, and there is a formula to determine that. If we are probing for the surface of a biological membrane, for instance in the placenta or in the lung, the Stereo Investigator software will generate virtual line segments, and we count an event, which is the interaction of the line segment with the tissue, with the membrane. The more intersections there are, the more surface there is. Today we're going to concentrate on estimating the number of cells in a given anatomical region. This in red is the virtual geometrical shape. It has to be a three-dimensional system because we need to find the leading edge of the particles. We could also call that the top of the cell. So we'll have a three-dimensional space, which is a cube or a three-dimensional rectangle, and that is called a dissector. And we'll focus down through the dissector, and when we encounter the top of the cell, if it is in the dissector, we count the cell. So the event when we are estimating the number of cells is the top of the cell in this special three-dimensional space that we call a dissector. Okay, but we are not only just going to count these cell tops, we are going to count them in a way that avoids bias. So we have to make no assumptions about shape or size or orientation or distribution. So how do we take care of making no assumptions about shape or size? We take the volume of the cell and we dwindle it down to 1 point. In other words, we're focusing down through the tissue, and as soon as the top of the cell comes into focus, we stop focusing and decide whether to count that cell based only on its cell top, or you could use the nucleus top, or you could use the nucleolus if there's one and only one nucleolus. So by dwindling the volume down to as close to a point as we can get it, points have no shapes or no sizes, so therefore we get rid of any bias that has to do with, for instance, counting larger cells more than smaller cells. We also want to make no assumptions about the orientations of these particles, and by counting the cell tops, that takes care of that. So I am going to show a little movie here of a group of cells in a chunk of tissue, and we are going to simulate focusing down through the z-axis looking for the cell tops. Each one of these cells has a red circle on its top, and this is what I'm going to be showing you in a minute using the microscope. We're going to be focusing up and down through thick tissue sections, 20 or 25 or 30 microns thick, using thin Z-planes. So in the case of these cells, we're going to have to use oil lenses, and that's so that we can find the tops of the cells. So for instance, right here, here's the cell top. It gets a red circle. Every cell has one and only one leading edge. Every cell has only one top. So if we count the cells this way, we won't overcount the cells. Not only that, but we can come at them from any direction. So in that last movie, we were focusing through the z-axis. Now we're focusing up and down this way. So we've turned this cube on its side. Let me just speed this up a little bit. And I'll show when we get to the part where we're simulating focusing up and down, this time through the x-axis instead of the z-axis. So the leading edge is now on the side of the cell compared to where it was before, but the fact still remains that every cell has only one cell top. If we count the leading edge or the top of the cell, then we won't overcount cells. So how can we manage to minimize bias this way? Well, we need to have thin focal planes. So this shows an objective which is 10 times with a 0.25 numerical aperture, and we have a wide Z plane. We may not even be able to tell one cell apart from the other, let alone optically dissect the top of the cell. In order to be able to focus on and find the top of the cell, we need to use an oil lens when we're dealing with cells of typical sizes. The oil lens and the Kohler illumination for bright field will give us thin Z-planes. If you're using fluorescence, you may need to go to the confocal level to get thin enough Z-planes to find the top of that cell. We also want to make no assumptions about the distribution of these particles. We want to avoid doing things like sectioning through one section in the middle of the region and assuming that that is representative of the whole region. So in order to get a good snapshot of what's going on in the whole region, we're going to use systematic random sampling, both on the intersection and the intrasection level. So we use a random starting point, and here it is the third section, and then systematic from there on every fifth section. When the sections get put up on the microscope, the XY locations are systematic, but for every section we'll have a different random throw. So not only does every section have an equal chance of being selected for sampling, also every XY location on a given section has an equal shot of being looked at. Now, how do we go about getting the estimate after we've done our sampling? So here is a little sketch of a dissector. This cell and this cell would be counted because their tops are in the dissector. We need to use thick tissue, 20 or 25 or 30 micron thick sections. There are one method of getting the estimate is to take a density. So that would be the number of cell tops counted divided by the number of dissectors times the volume of each dissector. So you can see how we'd come up with a numerical density of where we looked. But now in order to get the estimate for the whole region, we have to multiply by the reference volume, and that can be obtained by fluid displacement or by capillary point counting or by tracing. But the point is, if you work with a density, you have to have a reference volume to multiply by that density to get the number of cells in the whole anatomical region. And so that's why we recommend instead using the fractionator method. Today I'm going to be using the optical fractionator. It will use this method. Rather than multiplying a density times a reference volume, we don't need a reference volume because we keep track of the volume fraction. We keep track of the percentage of the volume that we are sampling, take its reciprocal, and multiply by all the cells that we counted in all the dissectors. What goes into the volume fraction? The section subfraction, the area subfraction, and the height subfraction. Let's look at the section subfraction. Section 1 means the first section where your region of interest appeared when you are sectioning. We are going to be systematic, so we're doing every fifth section, and I'm going to talk about how to decide how many sections to use in a minute. We don't always want to start with section 1. We want to make a random choice 1 to 5. So if the random choice is 3, we would be sampling on these purple sections. Our section interval is 1/5. We take the reciprocal. We need to multiply by 5 in order to extrapolate out on the section level. Here is the area subfraction. It is the area of the cross section through the dissector. That's called the counting frame, and I'm going to explain the red and the green sides in upcoming slide. The area subfraction, or the percentage of the area that you're sampling, is this area of the counting frame divided by the area of one grid step, which is the area among the counting frames. Here it's something like 1/20th. Here is an XY view of our tissue. We're going to talk about the height subfraction, so we need to look at the side view. Your dissector height could be the same height as the thickness of your tissue, but there's almost always going to be artifact where the knife cut at the top and the bottom of the section. So the height subfraction is the height of the dissector divided by the average mounted section thickness, and you're going to see that's one of the steps I'm going to be doing during the demonstration with the microscope, is measuring the top and the bottom of the section in order to get this height for the height fraction. Okay, so here are the counting rules. This is a cross section through a dissector. The dissector has 2 red sides and 2 green sides. So when you have a cross section, you see 2 red lines and 2 green lines. If the cell top or the nucleus top or the nucleolus, if there's one and only one nucleolus, and today during the demonstration I'm going to be using cell top, so I'm just going to say cell top from now on. If the cell top lies completely inside the counting frame, it gets counted. If the cell top crosses the green line but not the red line, it gets counted. The red line trumps the green line. So let's look at some examples. We focus down through the dissector, and we're looking at a cross section which is called the counting frame. This is how you hope they all appear, because this is an easy one. The cell top is obviously in the dissector. Here is a cell top that's more outside the counting frame than inside the counting frame, but it gets counted because it goes through a green line. Here's the opposite situation. This cell top is more inside the counting frame than outside, but it goes through a red line, so it does not get counted. This funny shaped cell top curves around, it hits the green and the red line. It does not get counted. The red line trumps the green line. There should be a red X through it. Let's take a look at this cell top. When I put a dissector down next to it and we look at the counting frame, notice that this cell top does get counted because the acceptance plane of this dissector is up against the rejection plane of that dissector. Notice that this cell top, which does get counted here, does not get counted for this dissector. So we're being very rigorous in our rules, and we're following these rules and taking a lot of pain to make sure we only count the cells that belong in this volume fraction, so that when we extrapolate out, we don't, for instance, count twice as many cells as we're supposed to. Okay, let's take a look at the same group of cells that we looked at before when we found their tops in the z-axis and along the x-axis. This time we're going to add the virtual geometric shape called the dissector. It has to be three-dimensional because we have to find the leading edge or the tops of the cells. So rather than focusing up and down through this and marking every cell top, we only mark cell tops that are within this dissector. Again, we have to use thick tissue sections, meaning 25 or 30 microns thick for an experiment like this. We need to use an oil lens so that we have a thin Z-plane of focus so that we can do something like find the top of this cell. So we're focusing up and down, and we find a cell that looks like its top is in the dissector. We can turn it on its side to prove that that top is on the dissector. We're going to be focusing up and down, seeing a red line and a green line, and I'm going to demonstrate that. So rather than marking every cell we encounter, only mark the cells whose tops are in the dissector and don't go through the red line. Let's show an example. The formula is the reciprocal of the volume fraction times all the cell tops you counted in all the dissectors in all the sections. We said that three things go into the volume fraction: the section interval, the percentage of the area that you are sampling, and the fraction of the height you are sampling. If your section interval is 1/10, you need to multiply by 10 to extrapolate out. If your percentage of the area you're sampling is 1 to 200, we need to multiply by 200. What if your dissector height is 20 microns and your average mounted section's thickness is 60 microns? That's a 1 to 3 fraction, so we need to multiply by 3. If you counted 100 cell tops in all the dissectors in all the sections, the estimate is 600,000. That estimate will be dead on if you followed the rules correctly and if your cells are completely homogeneously spread out. But we know they're not going to be homogeneously spread out. So we also have to think not only about following these rules to eliminate bias, but also about how much sampling we need to do to get a good snapshot of what's going on so we can see if there's a difference between the mean of the control group and the mean of the experimental group or groups. Search the literature. If you trust the paper, you can use those parameters. If they talked about why they've used those parameters, why they think they're doing enough counting, you are the one who is familiar with the salient features of your tissue. Keep an eye on the peaks of the data. If there's a group of cells that you never want to miss, like a clump of them, don't make the section intervals such that you could miss those cells. It can be a good idea to talk to a mentor about why they are sectioning as they do. If you really need some extra help about how many dissectors and how many sections to use, this is a great paper to read by Slomianka and West. And we have incorporated this procedure in our software. Oversampling and resampling allows you to determine if you can get away with using less sections or less dissectors. You're almost always going to be doing a pilot study on a representative animal from the control and from the experimental group. For a pilot study, we shoot for a couple hundred events. That means a couple hundred cell tops that are in the dissector. So we're shooting for 150 to 300 cells. You can choose the section subfraction to get about 10 sections. On each section, we can set up the area subfraction to get about 10 dissectors. We can size that dissector in the X and Y, Z dimension to catch about 1 to 3 cells. That will give us a couple hundred cells. Then we can look at a formula called a coefficient of error, which is designed to gauge whether the distribution of the particles is homogeneous or more clumped. And the lower the CE is, the coefficient of error, the more likely the chance that you got a good snapshot of what's going on, and you'll be able to see a difference if there is one between your groups. So in summary, to eliminate bias, we have to use thick sections, 20 microns, 25, 30 microns, when we're looking at the cells that are around the size of a typical biological cell, like you're going to see me do today. We want to use the tops of the cells. We need to have thin focal planes, and for the size of the cells I'm looking at today, it means using an oil objective. We need to honor the red and the green sides of the dissector or the counting frame. To be efficient, we want to plan for precision. There's a couple different styles, different labs like different styles. We can help you choose one of those. It's a pretty forgiving probe in that your sections can be oriented any way you like, but we must use thick sections. Okay, I'd like to pause, and I see some questions are coming in here. One question is, can I perform the optical fractionator in 40 times air objective, that's 0.75 numerical aperture, considering my coefficient of error is acceptable? Those two things are not really related. You need the high power objective and the high numerical aperture, so you'll have thin Z focal planes, and that will allow you to find the tops of the cells. So I would not use a 40 times air objective. I would want it to be a 40 times oil objective. But you can test this out empirically by focusing up and down through your tissue and determining, can I see the top of the cell, or does it look just like one big flat cell to me? The fact that your coefficient is good does not excuse the fact that you did not use thin enough Z-planes to find the tops of your cells. Okay, we have one question that is asking about the coefficient of error. Which value is acceptable for a good study? Traditionally, through the literature over the years, people have tried to get their CEs down to 0.1. The lower the CE is, the more of a chance you have of having done enough sampling so you'll see a difference between your control and your mean. Okay, next question gets at a good point. When counting cells, can the optical dissector be only 5 microns thick? That is really pushing it. I would rather see an optical fractionator height of 20 microns. If you have 5 microns thick and you have a times 100 oil objective, in practice you might be getting maybe 1 micron of resolution in the Z. So theoretically, you have 5 focal planes through that 5 micron tall dissector, but it's much harder to determine whether the cell top is in a 5 micron dissector than in a 20 micron dissector. So I'm going to say no to that. Get it 4 times bigger, about 20 microns, so you can focus up and down, find the cell tops, make good decisions about whether they're in the dissector or not. Here's a good question that gets at no bias about distribution. Is the method appropriate to sample rare cell types? And the answer is yes. So let's say that you only have a handful of cells that actually is your cell type. You are going to have to make your fractions almost equal to 1. So you'll put your dissectors right next to each other so that you have an area fraction of 1. you'll look at every section, so you have a section subfraction of 1. We still want to include guard zones, so we're not counting where there's artifact, so your height subfraction will be less than 1. And then what you can do is scan through at low power, and when you see your cell type, go up to the lowest power at which you can see the cell type, you're going to be going through a lot of empty dissectors, so you can scan through at low power and then go up to the power at which has the oil lens and decide whether that cell is in the dissector or not. So yes, it is appropriate for rare cells. Okay, how do you determine the thickness of a section if it was cut at 60 microns but has shrunk over time? And it won't, it's not so much the time that will shrink these sections, although that may be a factor. It's bringing them through ascending concentrations of alcohol, 70% alcohol, 80% alcohol, 95, 98%. That will cause osmosis, a lot of water will leave, and you will get a lot of shrinkage. Now, that's a great question because I'm going to show you how I determine the thickness of these shrunken sections when I do the presentation. And the answer is we show it where the top of the section is, and we show the program where the bottom of the section is. We have a instrument called a Z encoder that keeps track of where you are in the Z, and that's how we get the thickness. Okay, here's an interesting question. How do you define the region of interest if you are interested in counting cells associated with blood vessels? For example, parasites stained with a marker that shows the parasites. So you have to know where you are looking at the blood vessels. So your experimental question might be asking something like, does the length of the blood vessels change as a result of the experimental manipulation in a certain region? And then you will stick to that region, and you can do an optical fractionator. It doesn't matter if these cells are next to blood vessels. You're only going to be looking for the cells. So you have to be able to define your region. All right, well, thank you for those questions. At the end of this practical demonstration, which I'm about to show now, we'll pause for questions again, and then I'm going to show a couple summary slides, and then I'm going to stick around afterwards if there are more questions and keep answering them. So thank you for your interest and thank you for your questions. And now hopefully the parts that I just went over about theory will help us understand this practical demonstration I'm about to do. I did this on the system shown here. It's a Zeiss Axial Imager Z1 microscope. It has a lot of electronic components. For instance, the shutter that allows the bright field light to come through is electronic. Our software talks to all of those electronic components and controls them. I'm using a Q Imaging Retiga color camera. This is a bright field system. I have a slide that's stained with a Nissl stain. I am not going to use 10 sections. I'm just going to use one section to give a demonstration. I'm not going to use 10 dissector sites on each section. I'm just going to use about 3, just so I can give you an example of how to actually find the tops of these cells in practice. We are also using a Ludo Electronic Products MAC 5000 stage controller, motorized stage, and joystick. There's another important instrument right here. Before I show you that, though, I want to mention that this is a bright field system. We also set up systems that can work with wide field fluorescence or structured illumination, like an apotome. And our systems couple at various levels with different confocal microscopes. Now, I do want to show you this instrument, which is a little too small to see in this view, so I'm going to click on it. We have a side view here of the Z encoder. That's an instrument that has a piston that makes contact with a bracket. When you focus up and down, this bracket moves differentially from the bracket that holds the body of the encoder. And this tells us where we are in Z. And this is partial answer to one of the questions. This is how we know the true thickness of the section. So let's see that right now. This is a pre-recorded demonstration, and here is our software Stereo Investigator 2017. The probes are organized by what they do: number, length, surface, volume. Relationship among particles is called second order stereology. I'm going to go to the workspace tab and get some useful dialog boxes. I'm going to get the macro view, which will show a bird's eye view as we trace from field to field. The Z meter shows where we are in the Z, gets information from the Z encoder. A couple of these camera dialog boxes are going to help us. The program is asking us for a reference point: x equals 0, y equals 0, z equals 0. So we need to be able to see a live image. I'm going to the Acquire tab and I'm going to click Live Image. When I click Live Image, we can see the image. Our camera histogram comes alive, and we have a little icon of a microscope in the bottom right to show you are in live image. Let me just show that again. Here is live image off, and here is live image on, so that we can find where we want to put our origin, our x equals 0, y equals 0, z equals 0. Let's make the image better by increasing the exposure and doing a white balance. Okay, now we have an image that looks pretty much like it does through the eyepieces. So let's move. I am going to be estimating the number of large cells in these deep cerebellar nuclei. I'm not going to try to differentiate among those nuclei. Here are deep cerebellar nuclei cells, and it's asking for a reference point. I'm going to put that right in the middle of the cross section. And if we look down here, it shows the coordinates x, y, z. And if I can do this right, I'm going to get right in the middle of the origin and at 0, 0, 0 on the lower left. So that's x equals 0, y equals 0, z equals 0. Everything that you trace is in reference to that point. I'm going to make this a little darker so we can see the tracing a little better. And let's go up to the probes menu. And before we get number, I'm going to show you that we have a length probe called Spaceballs, which can be used to estimate the length of blood vessels or nerve fibers. If you want to estimate surface in a region, you can use isotropic fakir, which uses lines, surface of a particle. You can use the surfactor probe. If you want to estimate surface of a particle, you can use the nucleator probe. If you want to estimate, volume of a particle, and now we're on region, we can use Cavallari point counting. Second order probes look at the distance among particles. Let's go over to what we're doing today. We're not doing physical fractionator, which uses pairs of thin sections. We're doing optical fractionator. Notice that even in the icons here, we try to give you, tell you what's going on in pairs of thin sections for physical fractionator. We're using thick 20 micron sections. Okay, here's the optical fractionator workflow, and it is set up to have steps 1 through 11. And at each step, it tells you what to do down here. So step 1 is set up the subject with your name and the name of the slide. I'm just going to type demonstration. In the region below that, we can save the sampling parameters in step 9, and we can call them up here to use on all the animals in one group. Okay, this has to do with our section interval. I'm going to use an interval of not 8, but 10. We're going to look at every 10th section. There's 80 sections total, so that means we'll end up looking at 8 sections. These sections have been sectioned at 60 microns, but there's going to be shrinkage, and we're going to measure that. We don't want to start at section 1 every time, so we randomize the starting section. In this case, we're starting on section 2. Next step is set the microscope to low mag. So we really do try to have every step in here. We're already at a low magnification. Let's go to the next step. Trace the region of interest. We can name up to 99 different tracing lines or contours. I'm going to trace this cross section in the first section, which is section 2. I can be pretty loose when I trace the cross section. I don't have to hug the cross section, because I'm able to tell my cell type at high power. So I close the contour, go to the next step. We want to set the microscope to high power. We're going to need to use an oil lens. I'm going to bring us up to 100 times. In this Zeiss Axial Imager microscope, it dips down so we can load the oil. So we put the oil on, and we can see that the image is too blue. So we're going to do a white balance. And the image is not bright enough, so we can increase our exposure. Don't bring the exposure much past 100 milliseconds if you can help it. We can look in the macro view and see that we are in the region of interest. And now I'm focusing up and down. I'm in Kohler illumination. We can focus all the way to the bottom of the cell, all the way through it, see it is our cell type with a nucleus, and we can find the top of the cell. So we have enough thin enough Z-planes, thick enough tissue where we can optically dissect, find the top of the cell. So we're in good shape here. So we go to the next step. We're at 100 times. This is measure the amount of thickness. We want to refocus the top of section at each grid site because your section is going to be wavy. We want to measure the amount of thickness at each counting site. That's for the height fraction. But we need to get a first approximation of the thickness of this section. So I'm going to start taking measurements. It wants to know where is the top of the section. If you look to the right, we can see we are in the region of interest. Look even to the right of that, you can see the focus meter focusing up. We can see that we focused up to where everyone would agree we're out of focus, and then we focus down and we make a judgment call where the top of the section is. Those judgment calls will not vary with the control or the experimental group. We've controlled for all those types of judgment calls by doing unbiased stereology. So here's the bottom of the section. Again, that's needed for the height fraction. We have about 20 micron thick sections. Now we define the XY dimensions of the dissector called the counting frame. I'll make this darker so you can see it. So I'm in a certain place in my region of interest, and I see that I'm catching one cell. I'm going to move to another place in my region of interest. I'm trying to decide how big to make this counting frame. So I move over here, but there's none of my cell type over here. So I'm going to do another move from the Move tab with a Go To. And now I'm seeing that I would probably catch maybe 2 cells over here. So I'm thinking that 60 by 60 is a good size to make this counting frame. That's the XY dimension of the dissector. Okay, now we define the space between the dissectors. There's a couple ways to do that. I'm going to do it by saying, figure out a spacing that will give me 10 dissectors. It's a good way to set up a pilot study. Notice it shows you how many sites here. So 10 sites, 13 sites, 8 sites. It's always a random throw that you will not be able to control. I'm not going to use 10 sites. I'm going to use 3 sites. That will be enough to give demonstrations of how to actually find these cell tops. Okay, we can go back to step 5 and remember that we have just under 20 microns to work with. And that means I could use a 12 micron tall dissector. 12 minus 20 is 8. So we could have 4 microns on the top to protect us from counting where there is artifact. And whatever is left over will be for the bottom guard zone. In this next step, we can save the sampling parameters to be used on animals in the same group. For instance, 60 by 60, grid size. There's our Z information. Okay, this shows that we've drawn a contour for section 2, but don't forget we're going to have to do these other 7 equally spaced sections. We click Start Counting. The motors move for us. We can see in the macro view this random start point, and the dialog box asks us, show me the top of the section. That's for the height fraction. So we focus to where everyone would agree we're out of focus. We come down and we say OK when I don't think it's quite there, but maybe there. I can see some rips in the top of the section. That's the top of the section. Now we focus to where we're out of focus on the bottom. We come back up, and then we make our last motion downward, and that's the bottom of the section. And on the right, we show you a Z map, and I can make this a little brighter. And now we take a look at the cells that may or may not, whose tops may or not be in the dissector. This cell's top is up here someplace. It's too high. It doesn't get counted. Let's look at this cell. So we focus up and down. You can focus all the way through it to kind of make sure it is your cell type. There's a nucleus, a couple nucleoli. Focus back up above it. Now we need to focus down. You can see on the Z meter we're going down. I need to find the top of the cell. This is a judgment call that won't vary with the control of the experimental groups. I think there's a ridge along the bottom of this cell that looks like it's in focus. Maybe a little bit here is in focus. Look under the red line. It's not in focus. The top of this cell is in the X and Y and Z dimensions of the dissector. I don't want to use that dot though. I want to use this X that's labeled Nissl. I mark the top of the cell because it's in the dissector. It's not going through the red line. Let's look at the cell up top. I can go through the whole cell, make sure it is my cell type, and then focus up. I'm looking for the top of the cell. Now I'm focusing down, and I think the cell top is a ridge right around there. It goes through the green line. I count the cell. Now look at the macro view. We're moving to the next systematic random location. We're at a part of the nuclei that don't have any of these big cells. It doesn't matter that I surrounded them with the contour. We can just not count any cells. In other words, you can make this contour as loose as you want as long as you can tell the cell type at high power. So none of our cell types are there. Let's go to the next systematic random location. Now note this is a case where our cell type is actually outside of the contour that we drew. You can see in the macro view. So I'm marking the top and the bottom of the section, and just because it's out of the contour that we drew doesn't mean it's not our cell type. Here's the top of the cell. I can focus up. Notice the dissector turns yellow when you're in the guard zone. It's red and green when we're in the dissector. I can see that the top of this cell is not in yellow. It's in the red and the green. It's within the X and Y. It's not going through a red line. I don't care that it's outside of my contour. It's one of my cell types. I count it. Here we are in the fourth location. This happens sometimes where part is in focus. So this is out of focus and this is in focus. So I'm simply just going to pay attention to this bottom right quadrant on the top of the section and on the bottom of the section. So I'm deciding whether I'm at the bottom based on this region here, and I think that's the bottom. And on my Z meter, you can see the map. Now we have a cell there and a cell there whose tops are too high. Here's a cell. Let's focus through it, make sure it's our cell type, focus above it, focus down, find its top. It's in the dissector, it gets marked. Here's a cell over here, we'll focus above it, focus all the way through it, make sure it's our cell type, focus above it again. Here is its top, it goes through the green line, it gets counted. So this is why you don't want too many cells in a dissector, because it will kind of drive you crazy trying to remember if you have done that cell or not. We have done all those cells. Here's something over here. Right here, I'm focusing up and down. I don't see a nucleus or a nucleolus. I can't call that a cell. Maybe some kind of artifact or something, but I'm not going to mark it. Okay, so we go to the next site, and it tells us all of the sampling sites have been visited. If it was a pilot study, it probably would have been more sites. I like this. It prompts us to save after each section. And now we've done section 2, we've done the probe run. We need to find section 12. We need to go to the next section, so we need to find the slide that has section 12 on it. I can go to low power. The Zeiss software is telling me wipe the oil off the slide, but I don't really have to do that because the low power objective has a long working distance. Here's a shot of what we just did at low power. How could we ever have found those cell tops at this power? We can't. If you count at this power, you're counting cell pieces, not whole cells. Okay, so here is the next section, the second section, which because of the interval and the random start of section 2 is section 12. We did section 2, now we're on section 12. I go to the same nuclei. I can make this image look better. I'm going to focus. I'm going to decrease the light a little bit, and here's another way to do a white balance. And now I have an image that looks a lot like what I'm seeing through the eyepieces. Now I'm ready to go to section 12 because this is section 12. Begin next section. Is this the right slide? Yes. It's telling me I'm going to redirect you to the top of the workflow so you can do the same thing you just did in the other section. Back up here, say OK, and it brings me up to step 2. We're already at low magnification. Next step is trace your region of interest. Use the same contour. Again, we can be loose. We don't have to hug the cross section because we can tell the cell type at high power. We can even include parts of these nuclei which don't have our cell type. Close the contour. The next step is set the microscope to high mag. There's a little bit of oil left on the ×100 objective. I put a little bit more oil above section 12. We go into the oil and take a look at the macro view. It's showing us that our region, that our field of view is outside the region. So I can go to the Move tab, do a Go To, get ourselves somewhere in where these cells actually exist. Click there. Okay, now I see the cells. I need some more exposure time to get a brighter image. I'm not going to worry too much about the color because all I need to do is focus up and down, find the tops of the cells, decide if they're in the dissector or not. We've already done section 2. There's the contour, there's the probe run. We can collapse that back up. We need to click on the region of interest for the section we're in and click Start Counting. The motors take over, bring us to the first systematic random location in this second section. Same as we just did in those 4 examples, focus to the top of the section. That's for the height fraction. Focus to the bottom of the section. By focusing on the top and the bottom, we get the thickness, and we'll take the mean to be used in the height fraction. Those 2 cells are not our cell type, but there is a cell in the upper right corner, and we can focus through it just to make sure it is our cell type. I think that's its top. It does have a nucleus. Focus back up. Where the dendrite begins and the cell body ends is your judgment call. Again, that won't vary between control and experimental groups. We have controlled for the types of biases that could vary between control and experimental groups. Okay, we haven't done a whole pilot study. We're going to go to the next section. But this is a demonstration, so I'm not going to shoot for 100 or a couple hundred cells, which I would get if I went and did all of those sections. Let's go to step 11. I look at the results. They are organized by section. If you do open probe, you can audit and revisit all the sites. Use your shift key, pick all the sections, view result. It reminds us what the interval was. If we click here, it shows all the parameters that we use. For instance, if you remember 60 by 60 counting frame, this shows everything that actually happened in the probe. So this shows the number of cells counted and the thickness measured at each site for the first section. And then for the second section, we only did 1, counted 1 cells. That's the thickness. We give the thickness a couple different ways, and that means we actually give the estimate a couple different ways. We've only counted 6, but if we had counted a couple hundred, you could use the number weighted estimate. Our CEs are high. We wouldn't even look at them until we have a couple hundred cells. Here's the Z depth histogram. It bins the Z of that 20 microns of the section, and it shows you where you counted your markers, and this can help you set your guard zone. The bottom line here is we need more than 6, but use the number weighted section thickness estimate to get your estimate of the number of cells. So now we're going to pause and answer these maybe more questions about practical things, although we'll answer questions about theory also. Then I'm going to show a couple summary slides, and then I will stick around to answer more questions after. Okay. When counting fluorescence, there's a chance of bleaching the cells. Is the program and probe optimized to make the effect of bleaching factor negligible? That's an excellent question. The advice I give is if your fluorophore is robust, go ahead and do it live because bleaching is not happening unless you want to archive your data by taking an image stack, or if you have more than one color channel that you need to look at. For instance, you're counting double labeled cells, then it's a good idea to take an image stack of, say, maybe the red and the green channel so they're superimposed on each other. If there is bleaching happening, then it's a good idea to take an image stack. We do have built into the program something called systematic random sampling image stack acquire, and there's a workflow for that. It allows you to set everything up ahead of time, for instance, showing it where the top of the section is at each place where we're going to take the image stack, and then there's an automated process where it will take the image stacks for you with as many channels as you want, and then you can actually have your shutter closed and not be bleaching anything and work on the image stacks themselves. This can also free up the system that has the hardware. You can move to a desktop system and let somebody else collect image stacks. So the answer to that is yes, we do have ways to avoid bleaching, and that is taking image stacks. Okay, here's a good question. If there are no cells in the optical fractionator, why do you still have to measure the thickness? Well, you don't have to. And I could have omitted measuring the top and measuring the bottom. I could have just went right ahead and skipped that section without the cells. But it certainly won't hurt to measure the section thickness there. And if you have a lot of variance with your section thickness measurement, you may want to go ahead and measure it even at the places where there are no cells. So we get a good section thickness and then a good height fraction, and when we take the reciprocal, a good estimate. So here's a question. Is a high CE okay when counting cells? For example, cells that are more dispersed rather than closer together. And the answer is no. You want to get your CE low, both for cells that are very evenly spread out or cells that are heterogeneously distributed, like the question is talking about, in clumps. And you will have to do more sampling to get the same precision level if the cells are heterogeneous. So the idea is if the cells are more heterogeneous, put more dissectors down, take more sections compared to if they were evenly spread out, because we need more sections and more dissectors to actually find these cells. So you want a low CE whether they are heterogeneous or whether they're homogeneous, but it will be less work to get the low CE for the homogeneous situation. Okay, here's an excellent question. Doesn't having a loose contour, remember I was talking about I wasn't taking a lot of pain to hug the cross section of the nuclei where I'm trying to estimate the number of cells. Doesn't having a loose contour of your region increase the CE by having you sample extra sites that have no cells of interest? And the answer is no, that will not increase the CE. Kind of a ridiculous extreme example is you could make that contour as big as the whole brain. You would be going to a lot of dissectors that have 0 cells in them, but the formula is the estimate equals the reciprocal of the volume fraction times all the cell tops you counted. And if you add 0 to those cell tops, it will not affect your count. So it won't be as efficient if you have a very loose contour, but it's not worth the trouble of drawing a really tight contour unless you have to use the cytoarchitectonics at low power to be able to draw the region. Then you need to do a good job drawing that contour, really hug it at low power, because you're going to be counting on that line at high power. But what I was doing today, it was easy to see our cell types from the surrounding places where we weren't in a cerebellar nuclei, so I could draw it kind of loose. There's one other exception. We will give you a volume figured out by geometry in our program, and if you are using that, then you need to do a good job drawing around your regions of interest. Okay, this is a great one. When would it be best to also use the nucleator probe to obtain information about cell size as well? Okay, so the nucleator probe is what's called a local probe, and the optical fractionator probe is what's called a global probe. The nucleator probe is a probe that will give you an estimate of the volume of your particles. So while you're doing the optical fractionator probe, in fact, the optical fractionator probe will pick the cells for you without being biased by picking bigger cells more than smaller cells. The way we used to get volumes of cells was to look at their cross sections wherever they happen to get cut by the knife, and right there you're biasing yourself to looking at the bigger cells more than the smaller cells. So use the optical fractionator to pick the cells whose volume you want to get. That will get rid of any bias, and then we can focus down on the nucleus, put a point on it, and the program will shoot out rays, and you mark where those rays intersect the plasma membrane. It will stick it in a formula and give you an estimate of the volume. So you can get volume estimates while you're getting optical fractionator estimates, but for the nucleator, for the volume estimates of particles, you need to worry about how those particles are oriented. So we need to use a vertical or isotropic sectioning. Unlike the optical fractionator where we could use any types of sectioning we want, contact me about what to do if you don't use the vertical or isotropic sectioning, or even if you want to know what an isotropic or vertical section is. So yes, you can do the nucleator while you're doing the optical fractionator. Okay, here's another question. Can I measure my section thickness manually before, or will it be more accurate if I count it as I go? You certainly can measure it before. I don't know if you noticed when I was doing the optical fractionator workflow, but there actually was a checkbox. I checked the one that said measure the thickness before counting right now, so I could do it in front of you. But if you had already measured that section thickness at a couple different spots and wrote it down and you were happy with the variance of it, there was another checkbox I could have checked where I could have just entered that section thickness. So the answer is yes, you can measure it beforehand and write it down and then enter it into the program. Okay, here's a question. For fluorescent labeled tissue, do we have to use confocal? I'm just going to kind of paraphrase this question, or should we consider an apotome? So the example I gave today was bright field, but we can definitely set up systems that talk to fluorescent microscopes. If you're using just regular fluorescence, that's called wide field, and what you need to do is go to high power, focus through your cell. If you have many focal planes and you can find the top, go for it. If you can't, then you may need to go up to the level of an apotome, which is called structured illumination. That will give you crisper images, and what we're shooting for is to have many focal planes through the cell. And of course, a confocal is a great thing for that. That will give you very thin focal planes. The confocal and the optical fractionator workflow are made for each other in a lot of ways. Here's a question. If the cell was located outside the tracing line or the contour in the should you count that cell? And the answer is, if it is your cell type, yes, you should count it. So I was making my tracing contour kind of loose around the cross section through the nuclei. And you saw, even when I made it kind of loose, when I went up to high power, I was outside of the contour, but there was still one of my cell types there. So you have to use your anatomical knowledge to decide, is that my cell type or not? If it is, it doesn't matter if it's outside the contour. That just means you drew it wrong at low power. And if you're seeing all of these outside the contour, like more than a field away, then you really drew that contour wrong and you need to go back and draw it again. Okay. In summary, we are going to follow rigorous rules in order to avoid bias. We are going to suggest that you describe how you determine your region of interest in a rigorous way. If you do that, other people will be able to reproduce and verify your research. What do you need to let them know in the paper so they can do that? Let them know that we used the optical fractionator, that we used an oil lens times 100. Our section sampling fraction was 1/10. Let them know the percentage of the area that was being sampled. What were the XY dimensions of the dissector? What's the thickness of your tissue? What is the ratio of the height of the dissector to that tissue thickness? I read a lot of optical fractionator papers. The biggest mistake people are making is they're not doing it at a high enough power where they can find the top of the cell. Remember, every cell has only one top. We won't overcount that way. You want to report the average number of cells counted per animal and why you think you did enough counting. I want to thank everyone for your attention today. Please go to stereology.info to learn more about the optical fractionator probe, or if you can't get thick sections, you can learn about how you can use contiguous thin sections to do the physical fractionator probe. We can learn about probes for length, for surface, for volume, for second order. I would like to mention that there is an upcoming workshop in Europe. Here is the information for that. We also want to advertise Dan Peterson's workshop that's coming up, Quantitative Histology and Confocal Microscopy. It's a great course for unbiased stereology. If you want to watch this webinar again or send it to a colleague, give us maybe 24 hours and we'll get it up for this link. And if you didn't get a chance to write a question to me or didn't hear yours answered, I'm going to go ahead and personally answer all the questions. But if you think of another question, please contact us. Thanks a lot for your interest today.